present in the neck. It is an endocrine gland. Okay, the thyroid is the largest endocrine gland in the body. Okay, it was first described by Watson in 1659, who gave it the name thyroid because of its resemblance to because, of its resemblance to a shield. Okay. Mm, the thyroid gland is situated on either side of the trachea and just below the larynx. Okay, I will show you in the diagram. Okay, the thyroid gland is present situated on either side of the trachea and just below the larynx. Okay, the larynx just below the this here, just below the larynx. It's present in the trachea. It consists of two lobes. Okay, the two lobes are joined by a narrow isthmus connective tissue. These two lobes are joined by a narrow isthmus connective tissue. See here, this is narrow isthmus connective tissue. This is okay. The gland is composed of two types. Okay, this gland is composed of two types. One is follicular cells and second one is parafollicular cells. Okay, now let's discuss about follicular cells. These are circular cells lined by epithelial cells. These are circular cells which are lined by epithelial cells. Okay, and next, coming to the point, in middle contain colloid substance. If the colloid substance is high, the epithelial cells are in cuboidal in shape. Colloidal cells అనేవి ఎక్కువగా ఉంటాయి, epithelial cells అనేవి cuboidal shape లో ఉంటాయి. Okay. If the colloid is low, the epithelial cells are columnar in shape. If colloid is low, epithelial cells are columnar in shape. The colloid mainly contain thyroglobulin. Okay, this colloid mainly contain thyroglobulin protein. Okay, next coming to the point, this protein is source for thyroid hormone synthesis. This thyroglobulin is the main source for thyroid hormone synthesis, for the synthesis of thyroid hormone. Okay, remember it. Okay, the three types of hormones are released from follicular cells. Okay, there are three types of hormones that are released from follicular cells. The first one is triiodothyronine, then the second one is tetraiodothyronine, and the third one is reverse triiodothyronine. Okay. Now let us discuss about the parafollicular cells. Okay. These cells are occasionally occur in between basement membrane of follicular cells. These cells are occasionally occurs in between basement membrane of follicular cells. Okay, these cells secrete hormone calcitonin. These cells secrete the hormone calcitonin. Mm, it is involved in calcium metabolism. Okay, these parafollicular cells involves in calcium metabolism. Okay, let us discuss about the chemistry of thyroid hormone. Okay, see here, this is the structure of tetraiodothyronine. See here, this is the iodine is located in the third and the fifth position of the hexane component, and this is the structure of the tetraiodothyronine. Okay, the iodine is located in four positions. First are 3 and 5 places, and next 3' and 5' places. Okay. Hydroxyl group also attached here. So this is the overall structure of tetraiodothyronine. Okay, and this is finally they name it as 3 5 3 prime 5 prime tetraiodothyronine. Okay, now let us discuss about the triiodothyronine. Okay, here also 3 5 3 prime triiodothyronine. Here there is only in three positions the iodine is located. Okay, see here, the third prime and 3 5. Okay, third and 5 and third prime position, it is the triiodothyronine is iodine is located. Okay, here also hydroxyl group is present, oxygen is present, nascent oxygen. Okay, and finally this is the oral structure of 3,5,3' triiodothyronine. Next, let us discuss about reverse triiodothyronine. Okay, reverse triiodothyronine means, see here, 3,5 3' triiodothyronine. This is the chemical structure of reverse triiodothyronine. Okay, all thyroid hormones contain two phenyl rings. Okay, all thyroid hormones contains two phenyl rings. These are the phenyl rings. Okay, linked up by a ether bridge. Okay, these are linked up by ether bridge. Okay, now let's discuss. The two rings are outer and inner rings. Iodination at 3 prime 5 prime position of outer, 3 5 3,5 positions of inner rings in case of T4. Iodination at 3 5 position of inner, 3 prime position of outer rings in case of T T3. Okay. Mm, this is uh, okay. This is the chemistry of the triiodothyronine and tetraiodothyronine. Okay, an alanine molecule attaches at first position of inner phenyl ring. Okay, then hydroxyl group attaches at the fourth position of outer ring. Okay. Next, let us discuss about biosynthesis of thyroid hormone. Thyroid hormone synthesis requires two raw materials. Those are thyroglobulin and iodine. Okay. Now, thyroglobulin. Let us discuss about thyroglobulin protein. Okay. Thyroid hormones are synthesized by iodination of tyrosine residues of large peptide called thyroglobulin. Okay. These thyroid hormones are synthesized by thyroglobulin protein, as earlier I said. Okay, this is by which process it is synthesized means by the iodination of tyrosine residues of large peptides. Okay, thyroglobulin is a dimeric glycoprotein, contain nearly 5,000 amino acids. Out of these, only 115 amino acids are tyrosine residues, out of which 35 residues can be iodinated. After iodination, thyroglobulin is in colloidal state. Okay, thyroglobulin is a dimeric glycoprotein, contains nearly 5,000 amino acids. Out of these, only 115 amino acids are tyrosine residues, out of which 35 residues can be iodinated. 35 residues can be iodinated. After iodination, thyroglobulin is in colloid. Okay. Now let us discuss about iodine. The second substrate for thyroid hormone synthesis is iodine. Okay, the second substrate for thyroid hormone synthesis is iodine. Okay, um, daily requirement of iodine is 150 to 200 milligrams per day. Okay, its sources are drinking water, fish, cereals, vegetables, and iodized salt. Okay, thyroid hormones are synthesized in different steps. Okay, these thyroid hormones are in iodine synthesized in different steps. First one is iodine trapping, second one is synthesis of thyroglobulin, and third one is oxidation of iodide, and fourth one is organification or iodination of tyrosine, and fifth one is coupling, sixth one is pinocytosis and hydrolysis, and finally the seventh one is release. Okay. Now let us discuss about each one topic. Okay. First one is iodine trapping. Okay, thyroid follicular cells trap iodide ions only by actively transporting them from blood into cytosol is called iodide trapping. These thyroid follicular cells trap iodide ions by actively transporting them from blood into the cytosol is called iodide trapping. Okay, this iodide trapping occurs against this iodide trapping or occurs against concentration and electrical gradient. Okay, now let us discuss about the iodide concentration inside follicular cells is 20 to 40 times. Okay, it's 20 to 40 times, is 20 to 40 times. um The iodide concentration inside follicular cells is 20 to 40 times అన్నమాట. Iodide concentration follicular cells లోపల 20 to 40 times more than the of blood plasma. Okay, the anterior of the thyroid. Okay, see here. This is let us know about the anterior. Anterior means it will be like that's okay. Back. Mm, this anterior thyroid, okay, anterior of thyroid is negative because of negative charge iodide is present. Anterior of థైరాయిడ్ యొక్క anterior పొజిషన్ నెగిటివ్ గా ఉంటుంది ఎందుకంటే బికాస్ ఆఫ్ నెగిటివ్ చార్జ్ అయోడైడ్ ఈస్ ప్రెసెంట్. ఓకే, నెగిటివ్ చార్జ్ ఆఫ్ అయోడైడ్ అనేది నెగిటివ్ చార్జ్ లో ఉంటుంది వలన ఈ anterior ఆఫ్ థైరాయిడ్ యొక్క anterior పొజిషన్ అనేది నెగిటివ్ గా ఉంటుంది. In iodine trapping, the enzyme sodium potassium ATPase enzyme use ATP as energy. Iodine trapping లో సోడియం పొటాషియం ఏటీపీఎస్ ఎంజైమ్ అనేది యూజ్ అవుతుంది అనమాట. ఏటీపీ యాజ్ ఎనర్జీ సోర్స్ ఫర్ active transport. As ATP as energy source for active transport, so that's why we use the enzyme sodium potassium ATP is enzyme. And thyroid stimulating hormone strongly activates the iodide trapping. Okay, this thyroid stimulating hormone strongly activates the iodide trapping. Synthesis of thyroglobulin. Okay, thyroglobulin is a glycoprotein synthesized on rough endoplasmic reticulum, then enter into Golgi complex for glycorylation. Okay, thyroglobulin is a glycoprotein. This thyroglobulin is a glycoprotein as earlier we know. It is synthesized on rough endoplasmic reticulum. It is synthesized on rough endoplasmic reticulum, then enter into Golgi complex for glycorylation. Okay, sorry, excuse me. Then stored in colloidal substance involved in iodination. Now let us discuss about oxidation of iodine. Negatively charged iodide ions can't bind to tyrosine until they undergo oxidation to form active iodine. Negatively charged iodide ions can't bind to tyrosine. Okay, tyrosine to bind avavannamata negatively charged iodide ions until they undergo oxidation to form active iodine. Until until then they undergo oxidation to form active iodine ions. Okay. Mm, the thyroid is the only organ which can perform this oxidation step. This thyroid is only the organ which can perform this oxidation step. This is further catalyzed by the enzyme thyro thyroperoxidase. Okay, this oxidation of iodine is further catalyzed by the enzyme thyroperoxidase. Okay, the enzyme needs hydroxyl peroxide which is produced by NADPH dependent reaction. The NADPH is generated from the HMP shunt pathway. Okay, this thyroid stimulating hormone stimulates the oxidation process. Now let us discuss about organification or iodination of tyrosine. Iodination of thyroglobulin tyrosine residues is called organification, which is catalyzed by the thyroperoxidase enzyme. Iodine atoms now attaches themselves with tyrosine molecules. After this iodination of thyroglobulin tyrosine residues is called organification, as earlier we said, which is and it is catalyzed by the enzyme thyroperoxidase. Okay, the after this, iodine atoms attaches themselves with tyrosine molecules to form mm, okay, to form MIT and DIT molecules. Okay, now to form MIT and DIT. Here MIT means monoiodotyrosine, whereas DIT means triiodotyrosine. Okay. Mm, so monoiodotyrosine and diiodotyrosine molecules. The monoiodotyrosine and diiodotyrosine molecules still remain attached with the thyroglobulin protein. Iodination first takes place at third position, then the fifth position to form monoiodotyrosine or tyrosine and diiodotyrosine or thy- uh, tyrosine 2. Okay, thyroid stimulating hormone promotes the iodination of thyroglobulin. After iodine atoms attach themselves with tyrosine molecules, for what purpose means to form monoiodotyrosine and diiodotyrosine molecules. Okay, these both MIT and DIT remain attached with the thyroglobulin. Let now iodination first takes place at third position and then fifth position to form monoiodotyrosine or tyrosine and diiodotyrosine or dityrosine molecules. Okay, thyroid stimulating hormone promotes the iodination of thyroglobulin. This thyroid stimulating hormone promotes the iodine trapping. It also promotes the oxidation process and now this iodination process also. Now finally, now let us discuss about coupling. How the coupling process takes place. Okay, this coupling process is an oxidative process catalyzed by the enzyme thyroperoxidase. Okay, in coupling process, two molecules of diiodotyrosine fuse via an IEA ether bridge to form a molecule of thyroxine. Okay, the coupling process is an oxidative process which is catalyzed by the enzyme thyroperoxidase. Okay. In coupling process, two molecules of diiodotyrosine fuse. In coupling process, two molecules of diiodotyrosine fuse via an ether bridge to form a molecule of thyroxine. To form a molecule of thyroxine. Okay, tetraiodothyroxine. One molecule of alanine residues is released. As earlier we discussed, one molecule of alanine residue is released. Then alanine is converted into pyruvate and like pyruvate and amino group. Okay, NH3. This diiodotyrosine with the help of an enzyme, two molecules of diiodotyrosine fuse to form. See here the equation we given under this below. See, two molecules of diiodotyrosine fuse via an ether bridge to form a molecule of thyroxine. One molecule of alanine residue is released. Okay, then alanine is converted into pyruvate and amine group. Okay, with the help of an enzyme thyroperoxidase. Similarly, triiodotyrosine is formed by coupling of one monoiodotyrosine and one diiodotyrosine. Um, okay, see here I will show you in diagram the equation monoiodotyrosine and one diiodotyrosine fused to form triiodotyrosine and one molecule of alanine. Finally, this alanine converts into there is a single tyrosine and finally this alanine is converted into pyruvate and iodine group. In this process, deiodination takes place. Okay. Now let us discuss about phagocytosis and phagocytosis and hydrolysis. Okay, thyroglobulin is a storage form of thyroid triiodothyronine and tetraiodothyronine in their colloid. After stimulation of thyroid stimulating hormone on thyroid, there is a marked increase of microvilli of apical membrane. Okay, after the stimulation of thyroid stimulating hormone on hydrolysis and thyroid, there is a marked increase of microvilli. At, mm, of which membrane means apical membrane. Okay, this microtubule entraps the thyroglobulin into follicular cells by phagocytosis. This microtubule entraps thyroglobulin into follicular cell by which process means by phagocytosis process. Okay, these these phagosomes fuse with lysosome to form phagolysosome, in which various acid protease and peptidase hydrolyze their thyroglobulin into amino acids. including the iodothyronines. Okay, these phagosome fuse with lysosome. The In this process, phagosome fuse with lysosome to form phagolysosome, to which various acid proteases and peptidases hydrolyze the thyroglobulin into amino acid, including the iodothyronins. Release. Now finally let us discuss about release. After hydrolysis in large amount of tetraiodothyronin is released, then triiodothyronin is formed, by which process means deiodination of tetraiodothyronin. Okay. Inactive monoiodothyronin and diiodothyronin residues are deiodinated. Okay. Finally, in release, the inactive form of monoiodothyronin and diiodothyronin residues అనేవి deiodination జరుగుతాయి and reused for the synthesis of thyroglobulin glycoprotein, and this is also reused for the synthesis of thyroglobulins. Okay, thyroglobulins in the blood. Thyroid hormones bind with thyroxine. In blood, thyroid hormones bind with thyroxine binding globulin, transport proteins to reach the target tissues. This transport proteins to reach the target tissues. Okay.